So for this section of chapter 12, we're going to be talking about mutations, different types of mutations that can occur in the DNA in humans. So just as a review, a mutation is a change in the nucleotide base sequence of a gene or DNA. So we talked about all the letters that make up DNA, A, G, C, and T. We said that those letters show up in certain orders to make up a gene. You have codons of those letters, groups of 3 of those letters that make up genes. Sometimes different changes can happen when we're copying that DNA, and it can cause errors to take place. We call those errors mutations. Now, there are going to be a number of different types of mutations that we'll talk about. The key for this section is to know what those different types of mutations are. For example, when we talk about a germ cell mutation for number 1 on our list, this is going to be a type of mutation that occurs in the gametes or the sex cells, so in the sperm and egg cells of a human. When a mutation takes place in the germ cell of a human, it usually doesn't affect that organism itself. So say if you had a mutation that occurred in one of your germ cells, that mutation usually wouldn't affect you. What it would affect though, or what it could affect, would be your children. These are types of mutations that can be passed down to offspring. So germ cell mutations, they are usually going to be passed down to the offspring. For number 2, a somatic cell mutation, this is going to occur in the body cell of an organism. And this may affect the organism where the mutation occurs. For example, cancer. Let's take the simple example of skin cancer. If there were a mutation that someone had in their skin, they would develop skin cancer, and that would affect them very much so in their life. But something like skin cancer, that cancer is not going to be passed down to the offspring of that person, to the children. That's not something that would be passed down because it's not showing up in the gametes, it's showing up in that person's skin cells. So that's a big difference between those first two. Germ cell mutations, they do get passed down to the offspring. Somatic cell mutations, they usually do not get passed down to the offspring. A couple other types of mutations that we have on this slide. A lethal mutation, these are going to be mutations that will cause death in the organism, often before birth. Unfortunately, there are some mutations that can take place in the DNA of cells that might, kill off those cells before they even develop into a human being, or they might cause the death of a baby or a child, very early on in their life. Also, there can be beneficial mutations in all organisms. These are mutations that result in phenotypes or physical traits that are beneficial. We talked about some, mutations that might help organisms maybe blend in with their environment more. This can help them hide from predators and maybe help them, sneak up on their prey that they're eating. So there are types of beneficial mutations. They're not always bad. One of the biggest mutations we of course know is cancer. This is the uncontrolled growth of cells that can eventually invade other parts of the body. When cancer spreads, it gets more dangerous and more deadly. So for example, with skin cancer, if you catch skin cancer very early on, it's usually not that big of a deal. The doctor will remove that section of skin where the cancer is. However, if skin cancer goes on for a while and you don't know you have it, it can spread to other parts of your body and other organs, and that's when it can become very deadly. This is true for most cancers. When they spread, they become much more deadly. Good news, we're coming up with different treatments and different, ways of, not actually curing cancer yet, but ways of treating it and making it more of a livable condition for a lot of cancers. But there are still cancers out there that are still very, very deadly. It's something that we're going to continually be working on in our medical field. On this slide, we have examples listed of chromosome mutations. These are going to be mutations that you can actually see when you look at a chromosome. So the first type is called a deletion, and this is going to be a loss of a piece of a chromosome due to breakage. So a piece of the chromosome has broken off. That's usually going to be very, very bad for that organism because you're going to be missing genetic information. So we'll take a look at some things that can be caused by that. For number two, we have inversion, and that's going to be where a piece of the chromosome breaks off, flips around backwards, and reattaches. When you invert something, you kind of flip it upside down. So some of the genetic info on the chromosome has gotten flipped upside down, which can cause various issues. For number three, we have something called translocation. That's going to be where a piece of a chromosome breaks off and attaches to another chromosome, a non-homologous chromosome. So a piece of info goes from one chromosome, it breaks off, and it gets attached to another separate chromosome somewhere in the cell. So that can cause some obvious issues. And then for number four, we have one called non-disjunction. And that's going to be where chromosomes fail to separate during meiosis. So you might remember when we talked about cells splitting up during meiosis, the chromosomes have to split up too. They have to separate from their pairs. With non-disjunction, something happens where those two chromosomes do not split up. They get stuck together. And now we end up with cells getting extra genetic information and some cells missing genetic information. So that can be a very, very big mutation that takes place. This slide shows some examples of chromosome mutations. Let's start by looking at the strand in the middle towards the top where it says A, B, C, D, E, F, and it has the 4 arrows pointing away from it. This strand, we'll call it a chromosome, it has a couple letters on it. We can just say that these letters represent a gene. So we can say that this chromosome has 6 genes on it, okay? And remember, there are other, codes that make up those genes. So we'll just look quickly at what might happen and what it would look like if some of these mutations took place. So in the arrow pointing away from it on the top left, it says inversion. This shows us what it would look like if we inverted or flipped part of that chromosome. So we flipped around the genes C, D, and E. We flip them so that they now read E, D, and C. So this has changed the order of the genes on that chromosome. Okay, that can cause some big changes to happen. Let's move clockwise. Let's go over to where it says duplication, that arrow over to the top right. In this one, we have duplicated or doubled one of the genes. We added another D into there. So that's added a gene in there twice. So that, once again, could cause some big consequences for that organism. To the bottom right, we have insertion. And for this one, it looks like we've added another letter in here, another gene in here that wasn't there before. L. We added the letter L in there. So we've, once again, added extra information into that chromosome. That could be good, that could be bad. It just depends on how that information is going to be read. And then over towards the bottom left arrow, we have deletion, and that's going to be where one of those genes looks like it's been deleted. So C looks like it has been deleted from the original strand in the middle there. So those are some examples of what different mutations will look like up towards the top. We also have an example down on the bottom called translocation. So if we look at the left side, we have 2 bands of genes. These represent 2 different chromosomes. We have the one with the capital letters up on top and the one with the lowercase letters down at the bottom. If translocation occurred, then part of those chromosomes would be swapped. And that's what has happened. Up on top now, we have half of that chromosome that reads ABC from the original chromosome, but then the other half has been attached from the chromosome that was on the bottom. So we've taken a piece of the one on the top and we have attached it to the one on the bottom, and we've taken a piece of the chromosome on the bottom and moved it up to the chromosome on the top. This can happen sometimes in our cells. So that's why we're talking about it. Of course, that's going to really change up the information, and that could lead to some really big changes happening in those cells. So here's another picture that shows what some of these mutations could look like. On the left side, on that column, we have arrows pointing down showing the change that might happen to a chromosome if deletion took place. So we can see that we have two original chromosomes in the beginning, and as we move down, a little piece of the chromosome has gotten broken off somehow. So now we're missing information on that chromosome. Okay, that's not a good thing for that cell. For the middle column, as we move down, we have inversion. So we have a chromosome that has a piece that's been broken off and flipped around backwards. Once again, that can cause a really big change to take place. And then on the last column over to the right, we have translocation, where we have a piece of the smaller chromosomes, the two yellow ones, a piece of that is broken off and attached to the bigger chromosome. So that is an example of translocation. The piece of the chromosome is changed location. One of the biggest mutations that we've mentioned already is called non-disjunction. That's going to be where chromosomes fail to separate. They get stuck together during meiosis. The cause for non-disjunction is still unknown. It seems to be maybe like a random chance event that takes place, although there might be some genetics that goes into it and makes it more likely to happen in certain individuals. Um, it doesn't appear that anything that the individual does or doesn't do in their reproductive years can cause these changes to happen. So we're still trying to figure out why exactly chromosomes get stuck together, what causes it to take place. There are a few different types of non-disjunction that can cause different conditions to take place. The first type is one of the more common ones. It's called trisomy. Tri meaning three and somy meaning chromosomes. So this is going to be where an individual has an extra copy of one of their chromosomes. So in this case, they would have 47 chromosomes inside of their cells instead of the normal 46 that we usually have. So we can have different uh, versions of trisomy. One of the most common ones that we know of is called Down syndrome, and that is going to be where an individual has an extra chromosome 21. So their chromosome 21, they've gotten an extra copy of that. And of course, that leads to different uh, conditions in that individual that you might be aware of. It's like having, a little bit too much information for those cells. So sometimes that can lead to some mental delays and sometimes physical delays. There's Edwards syndrome, which is an extra chromosome 18. So we've had an extra chromosome 18 there. And then there's Patau syndrome, which is an extra chromosome 13. So there are a couple different versions of these, all of them that could have some consequences that show up for that individual, either mental or physical delays that can happen because of those extra chromosomes. So for Down syndrome, the extra genetic material by having the extra chromosome 21 causes delays in a child in the way it develops both mentally and physically, and it affects about 1 in every 800 babies. So this is a fairly common type of syndrome that's caused by a trisomy. So that's one of the important ones to talk about. A lot of us might know an individual who has Down syndrome. It is that common uh, in our population. For Edwards syndrome, that's having the extra chromosome 18. Unfortunately, most kids who have this don't live beyond their first 7 months. The girl in this picture shown is 6 years old, so some of them do live longer, but having that extra chromosome 18 seems to have more of a profound and harmful effect than having an extra chromosome 21. If they do survive having this extra chromosome 18, they usually have serious health problems because maybe their cells and organs just are not functioning properly because they don't know how to deal with that extra chromosome 18. So that's Edwards syndrome. Now, a monosomy, this is different than trisomy. Mono means one. So this is going to be where an individual has a deficiency in the number of chromosomes they have. They're going to have one copy of a chromosome where they're normally supposed to have two copies. So you'll have an individual when they look at their cells, they'll have 45 chromosomes in total, not 46. So when the egg's fertilized and where you're normally supposed to have 46 chromosomes, you'll have 45. So that you're missing a significant amount of genetic information. Monosomies are less likely to survive when compared to trisomies. Losing a complete chromosome is going to be usually much more deadly and much more harmful than having an extra one, even though that can be very harmful too, as we saw. A lot of times when an organism is missing a chromosome, that organism won't fully develop. So the cells that are starting to grow, maybe a human, those cells might stop developing because they don't have enough information to move forward. They might die. So usually monosomies are going to be very, very deadly for the cells. There are also types of mutations that affect only the sex chromosomes. So we have some sex chromosome abnormalities that might be caused by non-disjunction. These might be caused by chromosomes getting stuck together and an individual ending up with maybe an extra X chromosome or an extra Y chromosome. The first example that we have listed here is Klinefelter syndrome. And this is where an individual's going to have an extra X chromosome, even though they are a male. So instead of being XY, like you would normally find in a male, this person is XXY. They have two X chromosomes and then they have a Y chromosome. So they are genetically still a male, but that extra X chromosome may make some of the features of this individual more feminine, more like that of a female. Also, this might cause some issues with reproduction. This male might not be able to have children because of that extra X chromosome. They might have reduced facial hair. They might have reduced reproductive parts. So that would be some things that Klinefelter syndrome might cause. The second one is called Turner syndrome. And this is where a female is only going to have one X chromosome. They're missing the second X chromosome. With Turner syndrome, this individual, this female, might have issues reproducing because they only have one X chromosome. They might have issues with their egg cells when they're, trying to have a kid. So that's something that Turner syndrome could cause. There's a condition that sometimes is called, super males, having an extra Y chromosome. Remember, the Y chromosome is what makes a male. So if you have two of those, if you got an extra Y chromosome somehow from non-disjunction, then you are going to get an extra copy of that Y chromosome. So you might have more like testosterone, more of that male hormone. You might be growing to maybe a slightly taller height than average. You might have some other psychological issues that might come with that extra Y chromosome. So all of these come with different consequences, different conditions. That's the important thing to see. Any combination up to having 4 X chromosomes and a Y will produce maleness. So as long as there's a Y chromosome, that person is genetically considered a male. Males with more than one X chromosome are usually going to be underdeveloped and sterile. So that means that they might not be able to reproduce. Their sex cells are not going to be functional. Women that have an extra X chromosome, so XXX, or if they have if they're missing an X chromosome, if they're X and then like 0, they don't have the other one, those women are usually going to be sterile as well. So they will likely not be able to have kids. So those are some sex chromosome mutations that can happen. A couple different categories of gene mutations we want to talk about to wrap up this section. These are going to be mutations that happen inside of a gene. So the first type is called a point mutation. This is going to be either the substitution, addition, or removal of a single nucleotide, a single letter, and it occurs within a single gene or segment of DNA. So if you take one letter out, if you add a letter in, or if you substitute a letter, say if the letter is supposed to be A in this gene and you substitute it with a G, those are all examples of point mutations. They happen at one single point in the gene. So a substitution is going to be where one nucleotide replaces another. So one letter, say A, might be replaced by a C. That's a substitution. Sometimes we can have something called a frameshift mutation. That's going to be if some nucleotides are deleted or added in, the whole segment of DNA, the way it's read, is going to be moved. So not only does this change one gene, it might change a whole bunch of genes in the DNA. It's like taking one letter out of a sentence and then shifting all the letters over where that letter has been taken out. It can change the entire meaning of that sentence and really mix up the information. An insertion mutation is going to be where one or more nucleotides are added into the gene. And this also causes a frame shift. It's like adding an extra letter into a sentence and then shifting all of the letters of that sentence over. We've changed more than just one letter. We've moved all the letters in a direction where the entire sentence is different. So frame shift mutations, the point is that even if just one little change happens in the DNA, they can cause a frame shift to happen. And that means the entire DNA frame has been shifted. We've moved all the letters of the DNA. We've caused a big change to take place. This picture shows us an example of what can happen with these different types of gene mutations. Let's take the one on the top left that starts with CTT in the top left. That is the normal codon for a gene in DNA, CTT. If you remember from our last chapter, when we're going to make a protein from it, the first thing we got to do is we've got to make an mRNA message. So the normal mRNA message that we would make from that DNA, we're just matching them up with base pairs. Remember, C goes with G, and in this case, T goes with an A. So for this, we would get the mRNA message GAA. So we've taken CTT, and we've made the message GAA from it. And then we take that message, and we translate it into an amino acid. And when we looked at our chart last week, the message GAA would give us the amino acid called glutamic acid. That's part of a protein. Now, below this, where it says substitution, look what happens if we substitute one of the letters in DNA. Let's say a mutation takes place, and where it's supposed to read CTT, now it reads CAT, and A has been substituted in for that T. That not only changes the DNA, but it changes the mRNA message to GUA. So now we have a different message that we're using, and now that different message gives us a different amino acid. So that changes the protein. So just that one little change has made a big impact on the protein that we would be making. Now, on the next mutation here, we have another example. We have a longer strand of DNA that goes down through those letters. We have the message that we would make from it, right? The mRNA message. Remember, there's a U in mRNA, so A and U match up, and there's no T in mRNA. But we've base paired them up with A and U going together, and then C and G. And then from that mRNA message, we build these amino acids. We translate them into those amino acids. So we have 3 of them being shown in that middle picture. Now, over to the far right, what we have done is we've taken that DNA strand and we have deleted one of the letters. If you notice G, it was highlighted in the first strand. It has been removed from this DNA strand on this picture to the right. And now we have caused a frame shift mutation. All of the letters below that G have shifted up. So instead of the DNA reading ACG, now it reads ACC, and that gives us a different mRNA message, and that gives us a different order of the amino acids that are building that protein. All of the amino acids are different now. So now we're building a different protein. So sometimes adding in or taking away one letter can cause a huge change in the protein. And that can cause proteins that you need to be made not to be made. And that can cause really bad effects for those cells. So even just one letter change can be a huge difference in the DNA. That's why there's so many different types of mutations, because just one letter change can cause a whole bunch of different things to take place. So an example of a huge change in a protein that happens by a substitution error is sickle cell anemia. Here, in just one amino acid, we have a change of just one of the building blocks of a protein. This causes the cells in the person to go from the normal red blood cell shape, the round shape, into looking like the sickle shape that we see in the bottom picture of this slide. We see that one cell that looks sort of like a banana, looks sort of like a sickle. That cell is going to have issues in transporting oxygen and nutrients around in that person's body. Also, it's hard for that cell to fit into certain places in the veins of that person. So this can cause some issues called sickle cell anemia. So that's just one change in a protein that causes the cell to be completely misshaped. Okay, and many people actually have this. And this can lead to different conditions where you have anemia, where you have an iron deficiency, or where oxygen can't get around the way it's supposed to. So there is a discussion about whether sickle cell is a disease or if it's a trait. Normally, a person who inherits two genes, one from each parent, they produce the normal hemoglobin. This is a protein that's found in red blood cells. So normally a person gets two normal copies of that and they have normal red blood cells. A person with sickle cell trait inherits one normal hemoglobin gene and then one defective hemoglobin gene. This can actually be a good thing though, because this person would only have maybe a couple sickle cells since it's a recessive disorder, but it might provide some protection from malaria, which infects red blood cells. So being maybe a carrier for sickle cell trait, that might give a person an advantage over being infected with malaria. And a lot of people that have sickle cell trait, they live in these tropical areas. They live in places where malaria is common. So this is something that could actually be a good thing if you have just sickle cell trait. This gives you just a few cells that might have a sickle shape to them, but it causes a little bit of a change in some of the proteins on those cells that make it less likely to get malaria. A person who has full sickle cell disease inherits two bad copies of that hemoglobin. So this makes a lot of their cells sickle shaped, and this is where we have the bad consequences showing up, where they can't really get enough oxygen and iron around to their bodies. So there's probably different medications and treatments that they have to take throughout their life to deal with that disease. So some mutations and some traits are complicated, like it is with sickle cell anemia.